Enzymes are protein catalysts. Catalysts work to decrease the energy needed to start a chemical reaction, or it reduces what's called the activation energy. If we look at this diagram below, we have energy here. This is our reactant, our starting material, and this is our product, our end result, what we're making. There's this large hump in between the reactant and the product. In this particular case, the reactants have higher energy than the products. So this reaction should go, but there's this large activation energy barrier in between, which will keep the reaction from occurring. You can think of activation energy like if you have a piece of wood. Wood has energy in it, and you know that wood will burn. But you have to put it in a fire first, and then it burns. It needs to be activated. It needs extra energy. So an enzyme functions to lower the activation energy. It lowers the energy barrier and makes it more likely that the reaction will occur. Each cellular reaction has its own specific enzyme. An enzyme's substrate or reactant fits specifically into its active site. So let's look at this example below. First, we have an enzyme. It has an active site right here. And this is where the substrate will bind. Here's our substrate. In this case, it is sucrose. If you had a different reaction, you'd need a different enzyme. So this enzyme here is empty. It has its active site ready to go. The substrate comes and enters the active site, and as it does, it bends the molecule slightly. This can be thought of as a lock and key. Key enters, the enzyme, as the substrate enters, the enzyme kind of grabs it. Then water enters the reaction, and the substrate is converted into products. In the next step, the products glucose and fructose are released. And then the enzyme is ready for another round. It can interact with another sucrose molecule and split another one. To control cellular metabolisms, the enzymes have to be controlled. They need to be turned off sometimes and turned on sometimes. And there are two kinds of inhibitors that we're going to look at. One is a competitive inhibitor. The competitive inhibitor competes with the substrate for the active site. So this is an example of a competitive inhibitor. And this is the inhibitor. And what it's doing here is it's blocking the active site. The other kind of inhibitor is a non-competitive inhibitor. This kind of inhibitor alters the enzyme's functions by changing its shape. So this is an example of a non-competitive inhibitor. It doesn't bind or try and block the active site, but binds at a different location and changes the shape of the active site so that the enzyme will no longer function. Non-competitive. Okay. So there are two different kinds. the last thing we're going to talk about here is feedback inhibition. And this is very important, and you see it in many places in metabolism. Okay, so we've got the red molecule is our starting molecule. And we have enzyme 1 and reaction 1. And so the starting molecule, the red square, reacts and makes the B compound. And then we have another reaction, reaction 2, catalyzed by enzyme 2 to make C, compound C. Compound C, through reaction 3 and enzyme 3, makes compound D. So this would normally proceed if too much D builds up. If too much D is present, then we don't want to make any more D. So we'd like to shut this pathway down. D has the ability to go all the way back to the first enzyme and bind to it and stop the inhibition and stop enzyme 1 from working. So it will stop this pathway at the very beginning. It'll stop it right there. So we don't make B and we don't make C and we don't make any more D because we already have too much D. So that's feedback inhibition, and that helps to regulate metabolism, and there are many, many examples of feedback inhibition in all of metabolism. So many drugs, pesticides, and poisons are enzyme inhibitors. Many beneficial drugs are enzyme inhibitors. So enzyme inhibitors have been developed as pesticides and deadly poisons. So the ability to inhibit an enzyme can have a dramatic effect on metabolism.